Almost there. It's loading. And so give me only a few seconds and we are done. So welcome everyone. Today it's a new chapter of Neuroccino. It's 9:31 on Monday. And today is Lilith, who is going to present exciting paper, I think. And so we can just start. I will just allow you to share your screen. And okay, all yours.
Okay, hello everyone. So let me share the paper in the chat. Okay, so I'm gonna share the screen. Can you see the screen okay?
Yes.
Okay, perfect.
It's not full screen, but we can see the PDF.
Yeah, well, that's important. So yeah, today I want to discuss with you this paper called The Spacious Art of Single-Cell Genomics, published in PLOS Computational Biology last year. As you can imagine, this is quite not my topic, so it was a bit difficult to understand, but how did I end up here, you might ask. So the main reason is because Tara Akari and Lior Bachelor, they are challenging the methods that they use in their field, which happens to be the same method that we use. I'm talking about dimensionality reduction techniques such as linear principal component analysis and nonlinear methods such as UMAP and TSNIM. So basically, if you're not familiar with those methods, they are techniques that allow us to sort of zip our big high-dimensional data set into a smaller, manageable file that allows us to perform analysis much faster, efficient, and so on. Or you can think about it as condensing a huge book into several main points of that book, okay, that will allow you not get lost in all the details of the huge book. So this compression of the huge book into summary or zipping the file, okay, is called reduction, dimensionality reduction, or embedding your high dimensional large data set, okay, into smaller, lower dimensional dataset to, for example, 2D or 3D. So you can describe your dataset with, let's say, 3 or 2 vectors. Okay, so this is the methods that is a popular workflow pipeline in single-cell genomics because this field generates a ton of data. And larger data sets that I think neuroscientists could ever imagine. So that's why I think we can gain some insight from their work and see what they didn't like about these methods, what they don't like it. And this paper was published last year. So the way that they can use this method is one example, as I understand, is, for example, you have a cell from healthy tissue, okay? And then you have a cell, let's say, from cancer tissue. And each cell, you can look at different gene expressions in each cells, okay? And like this, we have, let's say, 2 data sets, one from healthy tissue and one from cancer cell. And then we can compress those 2 data sets and try to map it on the same 2D space. So we can visualize this as a map, okay, as a picture. And I think in case when these two datasets are mixing, you would think that, meaning that they don't create two different clusters. So I would think that probably they are different type of cells. One is cancer and one is healthy. But if they are in the same, probably they're healthy. So this is might be one of application. Another application can be so you can sort of track the development. So for example, you can treat one cell, okay, with a certain solution, and then see if that treatment, that condition makes some changes in gene expression. If there is some changes, then technically it should create another cluster on this 2D embedded space. So these are the methods that applications, and there are others that I don't think I should go into details. But these methods, they has to retain ideally local and global structure, meaning, for example, that we have two cells from the same tissue, okay, they has to cluster together in embedded space as well. So if we have something too similar in the original space, then we have to have the same relationship in the embedded space. Otherwise, we are violating everything, and we are changing the dataset and relationship between the cells and everything, right? So local structure is defined as nearest neighbor relationship. So if two cells are clustering in the same place or not. Is it clear? I hope so. So, yeah, and they said, okay, let's see if these methods, okay, they preserve after embedding local, global structure, and also the absolute distance between two cells. So let's go with the local structure. Some terminology they call ambient space, the sort of original space that had some, I don't know, some little preprocessing like normalization, for example. And you have like, I think it looks like this. Obviously, it's very, very simplified, but let's say we have like 4 genes and then we have like 5 cells, okay, but that's not the reality. And then each cell can have expression in some number. I think so. I don't know. But let's say we have this huge dimensional data set and we have embedded spaces. In this study, they tried 3 embedded spaces. One is a principal component analysis, with which they reduced their dataset to 50 dimensions. So they took first 50 principal components. Here, the second one is they did the principal component analysis. Third one, after PCA, they did UMAP. The latter two, honestly, I see it the first time, but it seems it's a popular pipeline in their study. Okay. So yeah, how would they understand if the local structure is preserved? So what they did, they take the ambient space, and within ambient space, they calculated the Euclidean distance between two cells. So for example, they took C1 and C2 and treated it like vector and compared and find the Euclidean distance. And they did it like, and they took the first 30 nearest neighbors to a cell, okay? And they did the same thing with embedded spaces. They took the first 30 nearest neighbors, okay? After that, they wanted to understand if those 30 neighbors are the same in the embedded space, okay? So they calculated Jacquard distance. Basically, it says how much these two batches are overlapping. So if the neighbors in embedded space are the same neighbors in the ambient space, the original space. Technically asking, is the local structure is preserved? So let's look at this plot. I hope you can see it. So on x-axis, we have Jacquard distance. Basically, Jacquard distance is calculated as 1 minus the overlap. So if the overlap is very small, then the number is big. It means that our batches are very dissimilar. There is no overlap. If the overlap is big, then the equation has a result closer to 0, okay? Means that they overlap a lot, so they are very similar. So the structure is preserved. In this case, you can see, as I said, closer to 1, we have better. We have closer to 0, we have better situation, okay? On the y-axis is the density, meaning that there are more cells in this region of Jacquard, in the range of this Jacquard distance, okay? As you can see, the situation is not so good, right? Because we have the average of Jacquard distance always greater than 0.8. So which means that the local structure in the ambient original space is not preserved in embedded spaces, those 3 embedded spaces. I said, okay, maybe it was because, right. So I thought maybe it's because, why I cannot see? Sorry. Yeah, there is a thing that is not allowing me. Okay.
It's Lilith gone? No. Yeah.
Yes.
It was me. Okay.
Yeah, she's
Okay.
disconnected.
Okay. Let's wait a few seconds. We also lost her a few minutes ago, so maybe she's coming back soon. Problem with the network. I will just let's hear. She's gone. Okay. Now it
Yeah, she
happened.
probably needs to close it and close Zoom and restart it again in order the screen sharing to work, I guess.
Yeah, probably she's doing that. I hope so.
I can dance if you want.
You have 15 minutes. Go ahead.
Yeah, make a start, Harren, make a start.
No dance. Neuro dance.
Lily's coming. She's back, I think. So we can continue the discussion. All about UMAPs.
Sorry, the
The
internet.
UMAP lab.
Yeah, the internet was yeah.
Yeah, no worry. Maybe you can share now your whole screen, so maybe we can see the PDF you wanted to show.
Yeah.
Yes, I need to make you co-host again.
Make me co-host.
Yes, done.
Okay. Let's see. Okay. Can you see it better?
Yeah, we can see the paper we shared before. I don't know if
Yeah,
you
perfect. So yeah.
Okay.
Yeah. So again, we see that the situation is not good because the Jacquard distance is closer to 1. So meaning that the local structure is not preserving. And I thought maybe it's because the PCA, maybe UMAP alone will do a better job. But in their supplementary material, they showed that even using 2D map and comparing it to the ambient space, you see it's not good either. Okay, there are different data sets with different sizes. Okay, the thing that they noticed is that the bigger is the data set, the worse is the local structure preservation. Okay, so this is something. And then they tried changing the neighbors, nearest neighbors, and perform different amounts of PCA, taking different amount of principal components in PCA. Nothing is changing drastically, okay? So yeah, so this is the first thing. The local structure seems, I don't know, based on this paper, is not preserved. Then they looked at the global structure. Well, they did sort of the same thing, but instead of individual cells, they look at the distance between cell types as a big categories, okay? But I don't know why they used in this case another measure to understand if there is like overlap between ambient and embedded spaces. But in this case, we have a Kendall-Stau correlation instead of Jacquard distance. And here, closer to 1 as a correlation, okay, then it means there is a similarity between ambient space structure and embedded space structure. But again, for example, with this TSNE and UMAP, it's not also the ideal over here, right? The authors are saying it might be not because of the methods, but because more reduction you do, more reduction steps you perform, more information you might lose. So that might be the reason. And I wanted to see in supplementary materials the UMAP alone performance, but I didn't find it. Maybe I should look at better, but yeah. Okay, so local structure is not okay. Global structure is not okay. And the last geometrical property that they looked at is the distance, the absolute distance. So over here, you can see a typical embedded map of your huge dimensional dataset into 2-dimensional dataset. Okay. And it's color-coded different types of cells. So they took one of the type, for example, chondrocytes and this, okay? They took this, and inside of this blob, they find those cells that are equidistant. So for example, this cell has the same distance between this and this, and between this and this one, okay? So they're equidistant. And within those equidistant cells, they divided them into those that are near and equidistant, so the distance is smaller, shorter, and the ones that the distance is far and equidistant. And then they say, let's now see where those dots, okay, located in the embedded space. And honestly, if you don't look at the title, you wouldn't tell which one is which, right? There is violation of the absolute distances between the cells. Okay, the distance is not equidistant anymore, and you don't really know if this one was the far or this one was the near. Okay, so again, seems that distance is not preserved either. Okay, so and they said this is not surprising because there is some lemma somewhere. Yeah, there is some mathematical lemma that that says that we need at least 1,800 dimensions with a 10,000 dataset cell to have only 20% error. So this is much more than 2 dimensions. So it is really not surprising that we would have a problem with absolute distances. So yeah, this is very surprising because basically we have nothing is preserved based on this paper, while at the same time UMAP, for example, is advertising, right? And also we see a lot of examples where it seems that there is preservation of local and global structures. Let's say those toy examples with the MNIST dataset, the numbers, okay? We can see that a sample of the number 2 is also close to another number of 2. So I cannot understand how to put this paper with the knowledge that I knew before. So yeah, I would like to hear your opinions on this. But then, yeah, there are a couple of things I want to tell. Well, here, I'm not going into details, beautiful pictures, but it's basically they are looking at how these distortions of embedding are affecting their practical applications within their single-cell genomics field. So I don't think this is relevant. Yeah, and maybe the last thing, and I think this is really puzzling, okay? They are trying to show that embedding is very arbitrary. And what they did, they created a new embedding technique. It's a joke example. They created new embedding technique called Picasso, okay? That method is trying to mimic qualitatively the well-established dimensionality reduction techniques. Basically, and how it works, they made it possible to, they developed an autoencoder framework to fit cells from any dataset to an arbitrary 2D shape. So it can be, the picture can be either elephant, it can be also in the form of flower, but qualitatively, they will perform in the same way as a UMAP, TSNE, or PCA. And you can see here, so we have the elephant, okay? And they calculated this correlation to ambient. And you see that elephant, a joke example, okay, is performing at the same more or less level as other methods. I don't know, this is to me, this is weird, feels weird. So I really want to discuss this and understand why and how. Okay. Yeah, and maybe the very last point is the reason why they chose this elephant. It's an allusion to a very famous story in the field of theoretical physics. So von Neumann said something like there is a whole story, but he said that with 4 parameters I can fit an elephant, and with 5 I can make him wiggle his trunk. So this was about the fact that arbitrariness is not a nice thing in science, even if you use just 4 arbitrary parameters. And then after that, they created a paper where they actually tried to use only 4 parameters to fit an elephant and make the trunk to wiggle. So yeah, and this is the paper. I hope you liked it. I hope you understood it, and I really need to discuss this with you. So yeah, if you have any ideas, how did this happen, or thank you.
Yeah, thank you very much, Lily. I think it's very interesting, especially because we are not all of us, but most of us, we're working also with this representation of the data by using this kind of approach. So I will let Chris to ask his question. So yeah, go ahead.
Hi, thank you for this paper. I'm gonna have to read it in more details. It's very interesting. One thing, so if I understand correctly, there is also a big influence on the dimensionality of the input data. So you, if as the dimensionality of the data increases, the UMAP and TSNE on their own become less efficient. So would there be are they advising like for a number, because here they talk about cells, but can we predict how much like if we need to use another dimensionality reduction before these techniques? Can we predict that before doing it, or do we just have to try with the different methods and see which one? Because also in that case they can test it because they have the output that they are expecting, but we might not have it every time. No.
Yeah. So this was this paper reminds me the paper when they took the dead salmon and they put it in an fMRI machine to show activation. It has the same vibe, I think, but I think they don't advise on a specific number. I think they more advise that these techniques we have to be very careful in interpreting the distances, the local structure, and they show that the map actually is very arbitrary. So I think they are more trying to say that this is very, so we have to try to avoid as much as possible those techniques in the certain applications, because
they only use the Euclidean distance for
so
UMAP.
they used also here they showed L1 L2, the Manhattan and Euclidean distance, and you see there is not so much difference when they calculate the distance between nearest cells, neighbors. I don't know, maybe it's the effect of larger data set as they said, but here they are not showing the UMAP alone again. Also, like I don't think it's nice to compare 50 dimension PCA with TSNE and UMAP, which is here, as I understand, just 2 dimensions. So also
there's been a paper, Valentina published a paper with a UMAP where I don't remember how she did it, but she determined that using 3 dimensions, a 3-dimension space was better, or 2-dimension was better. Like she used she had a way
MAE, we use the MAE, and but in my case was better. It depends on the data.
I think it might be it's just interesting to see if in lower dimensional data sets or smaller data sets, if this, to test it in our field, if this actually can affect, or it's about 904 cells. It's the smallest data set that they have. So I don't know.
To investigate.
It's something to look at it and be careful, and they said that this is just I mean, this Picasso model was, in the beginning I thought that maybe it's actual model. Like I was like, okay, interesting, well, and I was like, okay, but it's not performing the best, why are they suggesting? And then I understood that it was actually a joke.
Okay. And do they suggest something like to address this kind, like another kind of analysis, or just not to use it? Or
they are trying to say that the suggestion part I didn't have enough time, I think. No, actually I had to have, but I mean I didn't read so much in details these parts, but mainly, as I understand, is that they are saying that it's appealing to produce visuals, all that and all relationships, but we might distort our data like that, and maybe it's better to use more dimensions and calculate local distance and so on. Be more specific with your hypothesis rather than just, okay, let's put it all there and see what it will show into 2D map or 3D map. I think that's was their point, that maybe not delete totally these methods from your practice, but be careful, and maybe it's not necessary maybe to show the 2D or 3D, or you can show it, but it's not gonna, you cannot make any certain
Yeah, to be careful with
interpretations,
interpretation.
especially with distance.
Yeah. Okay. Eduardo?
Yeah, just a quick one. Thank you, Lilith. I think you already actually partially answered. And my question was if you have in mind some applications or things that could be translated in more specifically into our field, because yeah, I mean single cell genomics.
Yeah, well, technically you can reduce, because I mean we are using all the time as well these
Yeah,
techniques.
this yeah.
Um,
Because the problem seemed to me quite, but I also need to read the paper more in detail. It seems quite specific to the field, because here they say like we reduce hundreds of thousands of dimension to just 2 of them, which is a quite sharp reduction.
yeah,
And
but we sometimes we use their papers that we, for example, use PCA to look at the components instead of raw scores in your clinical data set, and then use the principal components to do the predictions of your create some machine learning algorithms, because PCA allows you to compress it and reduce the noise while retaining as much as variance as possible. So people are in our field also are using those methods. They are very popular. It's not specific to us in this paper, and I skipped that part because they start to talk about their own applications, but we might, try to take something from this paper and try to be careful in our analysis, I think.
sure.
Yeah, thank you. Ami, do you have a question?
Yeah, just very naive questions, but I thought that the point of these nonlinear dimensional reduction techniques are the fact that it's a nonlinear thing. But so if you measure distance based on kind of linear method, wasn't it the point? Like handwriting, like numbers, for example, they're not like if you just look at linear distances, they're the same number might not be close to each other, but there's some nonlinear dimensions that can kind of cluster them into distinct. So I'm wondering whether trying to measure the linear distance would necessarily map, but I guess the message is still clear that it's still arbitrary mapping, that if you're trying to measure the linear distance or any, I don't know, typical distance metrics.
Yeah, no, I think it's a very good question, because also I was kind of not sure if you can use Euclidean distance to calculate the distance between
so I don't know how
manifold space, okay?
Yeah.
But here it says that, for example, the Euclidean distance is the default metric for constructing neighbor graphs in methods such as TC and UMAP. So, as I understand, mathematically speaking, they use Euclidean distance for this math. So I really don't know so much under hood if it is mathematically plausible. I think it's more a question for a mathematician. But I had exactly the same thought. Maybe there is another metric that allows to calculate a distance in a different way that will retain those local structures. But yeah, I don't think you can use the same method of calculating the neighbors in the ambient space and those studies. I don't know. Yeah, but that's a good question. So.
Great. Thank you very much. I don't know if anyone has any questions. We are on time, so thank you, Lilith, for this very fascinating discussion, especially a topic that is very related to all of us. And for sure, we will have to read this paper to better understand how we can use these analyses to interpret of data. And yeah, that's all. I don't know if anyone has any comment or something to say. Otherwise, we stop here. And thank you very much for participating today, and have a very nice and exciting week in science, and see you next week. Bye-bye.
Thank you. Bye-bye.
Bye bye. See you next week.